My name is Dr. Jamie Caroon, and I am the Medical Director at the Center for Medical Cannabis Education in San Diego, California. In this video, I'm going to be talking to you about pain in the endocannabinoid system and how cannabinoids, both endo and phytocannabinoids, interact with the endocannabinoid system in the modulation and transmission of pain signals to and from the central nervous system. First of all, it's important to note that pain is not one thing. It is a heterogeneous problem. There are multiple types of pain. There are different mechanisms underlying those different types of pain, different etiologies or different causes. And in this way, pain is similar to cancer. Cancer is also not one thing. Prostate cancer can be dramatically different from breast cancer or lung cancer, and treatments for prostate cancer are not necessarily effective for other types of cancer. When it comes to cannabis, what should we expect? Should we expect that cannabinoids will be effective at treating all types of pain? Or maybe just certain types, maybe neuropathic pain or maybe inflammatory pain? And is THC more effective at treating one type of pain and CBD more effective at treating another? Are other cannabinoids more effective at treating certain types of pain? The truth is that we just don't know at this point, and research investigating cannabinoids as analgesic agents is really a work in progress. Pain can be categorized using various schema. Acute pain and chronic pain is one scheme. Acute pain is often thought to be adaptive because it's helping us as an organism understand that we have experienced a negative outcome by interaction with something, and therefore we should take measures to protect ourselves. And chronic pain is often thought to be maladaptive because the threat may not necessarily exist, but our bodies continue to act as if there is some sort of chronic problem. And another scheme that is often used in medicine bifurcates pain into nociceptive pain and neuropathic pain. And nociceptive pain is the more common type of pain that you see in clinical practice or that patients visit healthcare providers for. And nociceptive pain originates in damaged tissues outside the central nervous system. So it's pain that's induced by some sort of stimulus in the periphery, and that stimulus triggers a peripheral nerve to transduct a signal that makes its way to the central nervous system to effectively be interpreted as pain. Neuropathic pain, on the other hand, is pain that originates inside the nervous system itself. So this could be perhaps like diabetic neuropathy, where you have pain and tingling in your feet. And this is the result of nerves in that particular area experiencing usually an ischemic assault, meaning that they are not getting the oxygen and the blood supply that they need. So the problem exists in the nerve itself. It's not something outside the nerve. And so this is neuropathic pain. And diabetic neuropathy is an example of peripheral neuropathic pain. And multiple sclerosis is an example of central neuropathic pain. It's also important to discriminate between pain and nociception. It's said that pain isn't pain until it reaches the brain, and prior to that it's nociception. So pain is actually interpreted. It is a psycho-emotional experience. It's something that is perceived, it's subjective, and it varies from person to person. And it's defined as an unpleasant sensory and emotional experience that's associated with either actual or potential tissue damage. Preclinical studies, and these are studies that are conducted either in laboratory animals or in cell cultures and test tubes and petri dishes and things of that nature. Preclinical studies, they measure nociception, and then pain is inferred. When we want to understand whether a rat is experiencing pain, we obviously can't ask the rat, and so we interpret its behavior. And there's a lot more preclinical data than there is clinical data when it comes to cannabinoids and nociception and pain. And this is especially true with regard to CBD. There's more human clinical trial data using THC to treat pain than there is CBD. And we should keep in mind that it's possible that the anti-nociceptive effects that are seen in preclinical studies may not necessarily translate into clinically significant analgesic effects in clinical studies. In other words, what we see in animals may not necessarily translate to human beings. And this is an important point that we should not forget. There are 5 basic components of nociception, and the endocannabinoid system plays a role in almost all of them, really all of them except for conduction. So transduction is the first, conduction is the second, transmission is the transmission of that signal once it's been transducted and conducted from one neuron to the next. Modulation is what happens when neurons interact with one another to modulate that signal. And then finally, perception is how we experience the signals that we receive. And as I mentioned before, those are highly personal and very subjective. So we're going to take a look at each one of these components in the coming slides. Now we're going to review the neurophysiology of acute pain. And this would be a single, brief, noxious stimulus, which in this case is a nail penetrating the surface of the skin. So transduction begins with nociceptors of primary afferent neurons, which detect a noxious stimulus in the periphery. So a nociceptor is essentially a sensor. It's the terminal end of a sensory neuron that's picking up information from the periphery. And then afferent neurons send these signals towards the central nervous system. Efferent neurons send signals in the opposite direction, away from the central nervous system. So in this diagram, we can see that there's a nail that's piercing the skin of a finger, which leads to actual tissue damage. The noxious stimulus in this case is the nail, and it causes the release of inflammatory mediators from the damaged cells. And these mediators are histamine and prostaglandins and leukotrienes and cytokines and other things of that nature. And these inflammatory mediators act on the nociceptor itself and depolarize it, thus inducing a signal which gets conducted along the afferent neuron towards the pain centers in the brain. Now, there are 2 different mechanisms by which cannabinoid receptors inhibit transduction. There's a direct mechanism, and that is that CB1 receptors are on nociceptors. And when a tissue injury occurs, endocannabinoids are synthesized on demand, and they diffuse to nearby areas and activate those CB1 receptors on the nociceptor and effectively hyperpolarize the neuron and inhibit the transduction of the signal along that primary afferent neuron. So the cytokines act to depolarize the neuron, which would initiate that signal, and activation of CB1 receptors on nociceptors act to hyperpolarize or inhibit the sending of that signal along the neuron. The second mechanism is mediated by CB2 receptors, and this is a more indirect mechanism. CB2 receptors are predominantly found on immune cells, and these cells are often macrophages and mast cells that are in the periphery. And CB2 receptors, when activated, decrease the production and secretion of inflammatory mediators from these mast cells and macrophages. And in this diagram, you can see that there's a macrophage that is producing anandamide, which is an endocannabinoid, based on a chemical insult. That anandamide diffuses to bind to CB2 receptors, and there's also some CB1 receptors here, on the mast cell itself, or excuse me, on the macrophage itself. This is autocrine regulation because the cell is regulating itself. This leads to a decrease in the production of TNF-alpha and other chemokines. The anandamide also diffuses to a nearby mast cell where it binds predominantly to CB2 receptors, and it has the same effect, a decrease in the production of TNF-alpha. So it's decreasing inflammation through a receptor-mediated mechanism on immune cells. And as a result, these inflammatory mediators are being decreased, and therefore they're not able to depolarize the nociceptor here. So there's two mechanisms, one predominantly utilizing CB1 receptors, which is more direct, and the other predominantly using CB2 receptors, which is more indirect. Transmission is the next component, and transmission involves communication between neurons from one neuron to the other using neurotransmitters. And transmission occurs in three stages. We have the first order neuron, which is that primary afferent neuron that transducts and conducts the signal, where it synapses in the dorsal horn of the spinal cord with a second order neuron, which crosses the spinal cord and then ascends up the spinal thalamic tract to synapse at the thalamus, with third order neurons that project up to the pain centers of the brain, the limbic system, the somatosensory cortex, etc. So there are two synapses here, and this pain signal is transmitted from one neuron to the next at these two different synapses. Now, endocannabinoids, as we saw in the previous slide, are produced on demand. In the previous slide, we saw that they were produced on demand when there was a tissue injury. During neurotransmission, they're produced on demand when a neuron is depolarized. And unlike the traditional peptide neurotransmitters like serotonin or dopamine, which are produced in advance and stored in granules in the presynaptic neuron right here, endocannabinoids are synthesized on demand. They're lipids, they're not peptides, and they diffuse in the opposite direction. They move in a retrograde direction from the postsynaptic neuron to the presynaptic neuron, and they effectively act to shut it off. So when endocannabinoids bind to CB1 receptors on the presynaptic neuron, they suppress the excitation of that neuron and thus inhibit the transmission of that signal. So depolarization of the postsynaptic neuron induces the synthesis of endocannabinoids, which diffuse in a retrograde fashion to suppress the excitation of the presynaptic neuron. And that same exact process happens up here. And so this is how the endocannabinoid system is involved in transmission of pain signals. These CB1 receptors are on all the presynaptic neurons in each of these different synapses, and they are involved in this inhibition. The next component of nociception is modulation. These ascending pain signals are modulated along the way up to the central nervous system by descending neurons. These are descending modulatory pain pathways that run through a number of brain structures like the periaqueductal gray, the nucleus raphe magnus, and then also the dorsal horn of the spinal cord. These descending modulatory pain pathways can lead to either an increase in the transmission of a signal, which would be excitatory, or a decrease in the transmission, which would be inhibitory. And cannabinoids and cannabinoid receptors are intricately involved in this modulation. Endocannabinoids and CB1 receptors are found in high concentrations in each of these areas that I mentioned before, the periaqueductal gray, the raphe magnus nucleus, and the dorsal horn of the spinal cord. So just to take one example, if we look at the dorsal horn of the spinal cord, we have these neurons coming down, they're descending, and they are also having an effect at this synapse. In the prior slide, when we were talking about transmission, we were talking about the first order neuron and the second order neuron. This picture is actually complicated by the fact that there are thousands, if not more, neurons that are synapsing at this particular junction. It's just a chemical soup of neurotransmitters. But to simplify, these descending neurons are also releasing neurotransmitters that are acting on this synapse. And in the case of pain anti-nociception, they are acting to inhibit this signal. And so these are adrenergic neurons and opioidergic neurons and GABAergic neurons, etc. And they all have CB1 receptors. And they're all producing their own neurotransmitters that may be inhibiting that signal as well. And finally, there is perception, which is the process by which pain is recognized and interpreted by the brain. This is more psycho-emotional than it is physiological, but obviously the two are intricately connected. In pain management, it is often said that there is a difference between pain and suffering, and that both of them need to be treated. Suffering, as I mentioned before, is subjective. It's very individualized. Two people could experience the same pain, but interpret that pain very differently, and as a result, may suffer in a very different way. And suffering depends on these contextual factors, especially the person's individual interpretation. In the previous slides, we looked at these mechanisms and these neural pathways and the neurotransmitters and the receptors, etc. But here the focus is more on suffering, not nociception, and how cannabis may distort perception in such a way as to reduce the suffering by perhaps providing some sort of temporary distraction or inducing some euphoria such that the person doesn't feel as much suffering around the pain as they did prior to using the cannabis. There's one final point that I want to make here. This model that I just described of effectively the physiology behind the endocannabinoid system and nociception is based on agonism of cannabinoid receptors. And agonists are ligands or molecules that activate these receptors. And we produce cannabinoids, endocannabinoids, that are agonists for the receptors. And THC is also an agonist. But CBD is not. CBD works differently. So the model that was just described was really more about endocannabinoids and THC than it was about CBD. CBD also has anti-nociceptive effects, analgesic, anti-inflammatory effects, but the mechanisms are different. And to a large degree, they don't involve cannabinoid receptors. They involve other receptors. If you have any other questions, you can email me at jamie@karoon.com. Thanks for your time and attention during this video. Please check the website for other podcasts, videos, and blog posts in the future.